Welcome to this episode. Today, we're diving into a fascinating patent application filed just this past October by William and Dominic Carrington. It covers a novel class of synthetic cannabinoid analogs built on a completely new chemical scaffold, a xanthine core, that breaks away from the classical THC and CBD framework. This innovation brings a remarkable level of selectivity for the CB2 receptor over CB1, exceeding a 400-to-1 ratio, which opens up exciting therapeutic possibilities without the usual psychoactive side effects. So let's unpack the chemistry, pharmacology, therapeutic potential, and the development roadmap laid out in this patent.
Yeah, it's really a significant shift. The classical cannabinoids, like THC and CBD, are based on a dibenzopyrin core. But here, the Carringtons have introduced a dibenzopyrin or xanthine core. That's not just a minor tweak. It fundamentally changes the molecule's shape and properties. The increased planarity and polar surface area reduce lipophilicity, which means these compounds don't cross the blood-brain barrier as readily. That's key to avoiding CB1 receptor activation, which is responsible for psychoactive effects.
Right, and that's huge because CB1 activation is what typically causes the high associated with cannabinoids. So by structurally engineering the molecule to favor CB2 binding, which is mostly peripheral and immune-related, they've essentially redesigned the key to fit a different lock. The patent details 4 layers of structural modifications targeting specific positions on the molecule R1, R2, R3, and R6 through R8. For example, electron withdrawing groups like fluorine or trifluoromethyl at R6 to R8 reduce lipophilicity and limit CB1 binding.
And at R1, they use phosphate or acetate prodrug groups, which not only improve solubility, but also further reduce brain penetration. Thin, bulky groups like T-butyl or adamantyl at R3 sterically block CB1 binding sites. Plus, the amide-linked heteroaryl side chains at R2 orient the molecule toward the hydrophilic vestibule unique to CB2. It's a very elegant design.
The binding data really speaks for itself. The lead compound, Formula 9, has a Ki of about 12 nanomolar for CB2 and over 4,000 nanomolar for CB1, giving a selectivity ratio around 350 to 400 times. That's unprecedented in cannabinoid analogs. This level of selectivity is what makes the compound non-psychoactive and positions it well for clinical development without the regulatory hurdles that THC-like compounds face.
Exactly, and that selectivity opens the door to a broad range of therapeutic applications. The patent claims utility across several major disease categories mediated by CB2 activation. Neuroinflammation is a big one, suppressing microglial activation and reducing pro-inflammatory cytokines like TNF-alpha and interleukin-1 beta. Then there's cognitive recovery after neuroinflammation, with demonstrated improvements in Y-maze performance, which is a standard measure of working memory in rodents.
They also target neurodegenerative diseases, Alzheimer's, Parkinson's, multiple sclerosis, ALS, all conditions where neuroinflammation and microglial activation play a role. And chronic inflammatory pain is another key indication, with peripheral CB2 modulation providing analgesia without central nervous system side effects.
Don't forget mood dysregulation, major depressive disorder, bipolar disorder, treatment-resistant depression, where peripheral immune-brain axis modulation via CB2 could be therapeutic, and relapse vulnerability and substance use disorders, where CB2 modulation affects reward and stress circuits without addiction risk.
Beyond those, the patent outlines expanded possibilities in autoimmune diseases like rheumatoid arthritis and inflammatory bowel disease, oncology support through tumor microenvironment modulation, rare diseases like Huntington's and Rett syndrome, and even metabolic diseases such as type 2 diabetes and non-alcoholic fatty liver disease.
What's strategic here is the absence of psychoactivity. Because the compounds have a CB1 Ki over 10 micromolar, they're non-scheduled and non-psychoactive. That means they avoid the complex DEA scheduling process that cannabis-derived medicines face. This could lead to broader prescriber acceptance, including in pediatric and elderly populations where psychoactivity is contraindicated. Plus, there's potential for over-the-counter formulations in pain and mood disorders.
Moving on to drug delivery, the patent supports a wide range of administration routes. Oral tablets show about 38% bioavailability in rats, with a half-life around 3.5 hours. There's also intravenous phosphate salt solutions for rapid onset and 100% bioavailability, transdermal patches and gels for continuous delivery, subcutaneous depot injections for controlled release, and even intranasal sprays, sublingual fast-dissolve tablets, inhalation nebulizers, and intrathecal injections.
The prodrug strategy at R1 is particularly clever. Phosphate and acetate prodrugs improve aqueous solubility, which is critical for parenteral and oral formulations. They also allow controlled bioactivation kinetics, so you can tune the onset and duration of action. Plus, they reduce gastrointestinal irritation compared to direct phenolic forms. They even have pegylated and amino acid-linked prodrugs for extended half-life and targeted tissue delivery.
The oral formulation example is quite practical. A 172 milligram tablet with 10 milligrams of active ingredient, combined with microcrystalline cellulose, mannitol, hydroxypropylcellulose, and magnesium stearate. It's a standard pharmaceutical approach, showing they're thinking about real-world development.
On manufacturing, the patent provides detailed chemical synthesis routes for lead compounds, involving 6 to 7 steps with commercially available starting materials. Key reactions include Friedel-Crafts alkylation, aromatic bromination, cyanation, amide coupling, and phosphorylation. The overall yields are moderate, around 10 to 17%, which is typical for complex synthetic cannabinoids.
But what really stands out is the microbial biosynthesis approach. They've engineered Aspergillus niger to produce these compounds using synthetic geranyl-CoA transferase and prenyl transferase enzymes with CRISPR interference to suppress competing pathways. Plus, a CB2 responsive GFP biosensor provides feedback regulation of production. Current yields are about 21 milligrams per liter, with potential to scale up to 100 to 500 milligrams per liter through fermentation optimization.
That's a game changer for commercial production. Microbial biosynthesis can reduce costs and improve scalability compared to traditional chemical synthesis, especially for complex molecules like these.
The patent also lays out a comprehensive data generation framework. They've started binding affinity and selectivity assays using radioligand displacement, and plan extensive structure-activity relationship studies across substitution positions to optimize potency and selectivity.
In vitro pharmacology includes CAMP modulation, beta-arrestin recruitment, ERK phosphorylation, and calcium flux assays. They're also measuring anti-inflammatory biomarkers in macrophages and microglia, and neuroprotection assays like oxygen-glucose deprivation and excitotoxicity models.
ADME and pharmacokinetic data show oral bioavailability around 38% in rats and a plasma half-life of about 3.5 hours, with a target human half-life of 4 to 8 hours. They plan further studies on protein binding, brain penetration, designed to be less than 0.1 brain-to-plasma ratio, metabolic clearance, and cytochrome P450 inhibition.
The in vivo efficacy data is compelling. Formula 9 at 10 milligrams per kilogram orally reduces microglial activation by over 60%, TNF-alpha by 57%, INTERLEUKIN1, beta by 55%, and improves cognitive performance by 31% in the Y maze. They plan to expand dose-response studies and test in additional models of pain and neurodegeneration.
They also have addiction and relapse models showing attenuation of opioid and cocaine reward behaviors, which could be very important for substance use disorder therapies.
On the clinical side, the patent outlines IND-enabling studies including safety pharmacology, acute and repeat dose toxicology in rodents and non-rodents, genotoxicity testing, and pharmacokinetic and toxicokinetic studies. Phase 1 clinical studies will cover single and multiple ascending doses, food effect, and drug-drug interaction panels.
Phase 2 and 3 endpoints are tailored to indications, cognitive scores like MoCA and MMSE for neuroinflammation, pain score reductions for neuropathic pain, depression rating scales for mood disorders, time to relapse for opioid use disorder, and cognitive and biomarker endpoints for Alzheimer's disease.
The commercial landscape is substantial. Alzheimer's and dementia affect nearly 7 million people in the U.S. alone, with a global market over $13 billion. Chronic neuropathic pain affects about 20 million Americans, major depressive disorder over 21 million, and opioid use disorder around 6 million.
Competitive advantages include superior CB2 selectivity compared to prior agonists like GW842166X and JWH133, non-psychoactive and non-scheduled status avoiding regulatory hurdles, and potentially better side effect profiles than gabapentinoids. Oral bioavailability and half-life also support patient compliance.
Intellectual property exclusivity extends through 2045, with potential extensions and orphan drug designation for rare neurological diseases. That's a strong position for either independent development or partnerships.
To wrap up, this patent establishes a novel, structurally unique, and pharmacologically selective CB2 agonist platform. The non-psychoactive profile removes a major barrier in cannabinoid drug development. Priority next steps include completing SAR studies, full ADME profiling, expanded in vivo efficacy, IND-enabling toxicology, and Phase 1 IND filing within the next 2 years.
It's a well-thought-out platform with strong scientific and commercial foundations. Definitely one to watch as it moves toward clinical development.
Absolutely. Thanks for joining this deep dive. Until next time.
Take care.